Hello, thank you for that introduction. As we said, I am Abby, and today I'm going to talk to you about squids. So, yeah, I unmuted. There we go. Okay, so my dissertation is comprised of 4 pretty different chapters. The first one being, does RNA editing affect the use of transcriptomics in cephalopod phylogenetics? Followed by, what is the evolutionary history of ammoniacal buoyancy in egopsid squids? And then, what are the present and future niches of pyrotoothed squids? And finally, what is the biogeographic history of these pyrotoothids? So my dissertation is structured kind of hierarchically, starting with the superorder Decapodiformes, our largest clade, moving to the order Egopsida, and followed by the family Pyrotoothidae. So we're going to start with the superorder Decapodiformes for our RNA editing and phylogenetics chapter. Decapodiformes is a large clade consisting of 7 smaller clades. We have Myopsida, the nearshore squid, Egopsida, the open ocean or oceanic squid, Sepiolids, the bobtail squids, Idiosepiids, the pygmy squids, Spirulida, the ram's horn squids, Sepiida, the cuttlefish, and then Baphytoothioidea, a clade of deep sea squids. And so we have been trying to figure out the deeper order relationships between these orders. We know Decapodiformes is a clade. We know each of these smaller clades are a clade, but how are they related to each other? And we have found success in the past couple of years using transcriptomes to elucidate these phylogenies. And so this is a tree from our most recent transcriptomic paper, Lindgren et al. 2022. And these colors will represent the orders for the rest of this section. So you can see up here, Sepiida and Myopsida are large-bodied, shallow water squids are grouping together. And then Egopsida and Spirulida are the outgroups to those. They're sister. You've got our large-bodied deep sea squids, followed by our small guys, which is Sepiolida and Idiosepiida as sister to the rest of Decapodiformes. But cephalopods have this, all organisms have RNA editing. And specifically A to I RNA editing. And I'll talk to you about why this is important in cephalopods in just a second. So A to I RNA editing is when an enzyme called adenosine deaminases that act on RNA, or ADARs for short, they work on all of the A base pairs in their RNA to make inosine. So you start with a genome, you start with your DNA, and let's say the sequence is GTC. That sequence is then transcribed into CAG, and this is where that RNA editing happens. So you have the enzyme comes and changes this base pair to an inosine, which is then read as a guanosine. So before translation, the result, before RNA editing, the result of translation would be glutamine. And then after translation, it would be, after RNA editing, it would be an arginine. So it's physically changing the structure of the proteins that cephalopods can produce without having to alter that genome. And so in this presentation, we talk about editing sites versus recoding sites. So editing sites is any site in the genome where that RNA is editing. Recoding sites is when this happens and there's a change in that protein at the end. So in cephalopods, the RNA editing is a vastly higher rate than in any other organism that we have found so far, at least. Humans, mice, and flies, they have less than 1,000 number of recoding sites in their genome and transcriptome. But squid, 60,000. So we're seeing it's a way different number. And the fraction of proteins we're housing recoding sites, again, is almost 60% of all proteins have at least one recoding site in. So yeah, it's definitely affecting cephalopod transcriptomes. But is this just noise? Is this having an actual functional effect on these cephalopods? And Liskovich-Brouwer et al. 2017 did a study identifying these editing sites, and they looked at these potassium channels. This editing site is increased levels in the nervous system in cephalopods. And so they looked at this potassium channel closing time, and in squid, cuttlefish, and octopus, so 3 separate clades of cephalopods. And they all have a different closing time, you can see here. As soon as this protein is edited, they all converge on the same rate. And whether this is somehow buffering mutations, this is some ideal rate, and they can allow their genome to evolve. There's lots of mutations that are happening, and they don't need to control for those actual mutations in their genome. They can just edit the protein, and then you get the ideal rate. We don't exactly know why this is happening. We don't know if this is like the best rate, what is happening here. But we know there is some functional effect of this RNA editing. So why does this matter to me? I'm a phylogeneticist. Why do I care about RNA editing? It could potentially be giving us false signal. So let's say in our tree, we have Myopsida and Sepiida grouping together as sister. Let's say they have different sequences in their genome. We have DNA is saying GCC, and the DNA in Sepiidas is saying GTC. So if this was happening across the genome, those species would not be grouped together on that tree. But when you have this editing, there's no editing in Myopsida, let's say, and it's CGG, but you have an editing here, and now the resulting RNA that we're using to build the tree is showing that these two have the same sequence and are more closely related. And so ideally, we would use genomics to solve this problem. Genomics are more computationally expensive. Sequence are more expensive. They have a lot of these noisy regions like introns and things that are not expressed. So ideally, transcriptomes are the perfect candidate for phylogenetics. We just need to make sure that this is not happening in our data set. So I'm doing two main analyses. This regular versus corrected for editing site analysis, where I've downloaded the regular RNA-seq reads from GenBank, assembled those, and then I took those same transcriptomes, identified the editing sites, and treated those as missing data to compare those two trees. I am still working on these analyses. We hit some snags along the way, so I don't have those trees to show for you today, but I do have trees from the next analysis. So because these editing sites are highly enriched in the nervous system, I took transcriptomes from nervous tissue and transcriptomes from non-nervous tissue, so muscles, mantle, suckers, things like that, and I compared them for the same species. So it's the same set of species in both trees. And to compare the trees for these analyses, I'm comparing their topology, the branch lengths, and then their gene concordance factor, so a support value. For my regular versus corrected, I'm going to briefly go through this, even though I don't have trees to show you, just to show that I'm working on this. This is coming. I have 37 total genomes, or total transcriptomes, and I have 6, these ones highlighted in green, with their genomes. And that is important because of our pipeline. So in order to identify these editing sites, I took the Illumina data, so the reads and the corresponding genomes, mapped those transcriptomes to their genomes, used SAM tools to change file format, identified the editing sites using Ready tools, and then I mapped all of the transcriptomes that didn't have genomes to their closest relatives' genome. So let's say I have Sepia feronis and another one, individual from Sepia. I mapped those together to identify the editing sites, because what this spits out is the region of the genome where an editing site is. And I changed all those editing sites to N. But overall, our bioinformatic pipeline, just to build the tree. So once we have all of the transcriptomes assembled, corrected, whatever we're doing, I used the Pincho modular assembly. This is a really cool program that you can do some pre-processing, multiple assembly methods to build a consensus assembly, and then some post-processing. And I can go into details further if you are interested afterwards. I've identified the protein coding, the open reading frames in transdecoder, put it through OrthoFinder to find orthologous genes, aligned them, and then built quick maximum likelihood trees for all loci. Those maximum likelihood trees were put into PhyloPyPruner to identify paralogs and get rid of those in our analyses, and then filtered further to remove genes with high rate and composition heterogeneity. Fast concat concatenated all of my genes into one matrix, and then I used IQ-tree for maximum likelihood tree to get our phylogenies. So the results. These are the results for the nervous tissue and the non-nervous tissue. When I put these orders up, you are going to see a difference. In our non-nervous tissue transcriptomes, we are getting the topology that we got in our most recent transcriptomic tree. We have Sepiida and Myopsida as sister, followed by Agopsida, and Sepiolida and Ibiosepida are the sister to the rest, that large body plate. But in our nervous tissue transcriptomes, we are now seeing Egopsida and Myopsida as sister to each other. And this isn't entirely surprising. Other analyses have found this relationship. These guys are usually lumped together as the squids, the true squids. So they have morphological data that are supporting this relationship, but there's also morphological data supporting this. So this is what we're getting with all of the transcriptomes, all 30 plus of them. And then this is the interesting one when we use only nervous tissue transcriptomes. One thing to note, though, is that this is a slightly smaller data set. You have about 2,000 fewer orthogroups and around 200,000 fewer variable sites. Now, this is still a large data set. So we should be able to capture and get rid of all of that noise because it is such a large data set. But it is interesting to note that the smaller one is giving us the different topology. So overall, nervous tissue versus non-nervous tissue, different topologies. Is this an artifact of data set size, or is this actual RNA editing? When I finish my further analyses, hopefully I can kind of tease that apart further. And then finally, differences in branch lengths can affect downstream analyses. So there are slight differences in branch lengths across the different trees. So this could affect things like divergence time estimations, ancestral state reconstructions, things like that. So we've talked about the superorder Decapodiformes, and now we are moving on to the order Egopsida. Egopsida are those oceanic open eye squids, the ones I like to call the weirdos. They're my personal favorites. And we're going to talk about the evolution of this trait called ammoniacal buoyancy. Certain cephalopods can sequester ammonia, which is typically a waste product in most animals, and use it to maintain their position in the water. Ammonia neutral buoyancy is really important, specifically for these deep sea squids, because they want to maintain their position in the water column. They don't want to float up. They don't want to sink without using very much energy. There's not a lot of food out there. It's hard to find energy. So you don't want to expend a lot of energy to maintain your position. And ammoniacal buoyancy, ammonium is less dense than seawater. And so that, combined with the body tissues, is allowing them to maintain that position. Also, ammonia does not compress with depth. So that buoyancy, instead of things like the swim bladder, which is going to compress with depth, ammoniacal buoyancy allows these deep sea species to also remain neutrally buoyant. There is a variation in storage mechanism, which I will talk about in a second, but here you're seeing the arm cross sections of two ammoniacal squids, one that stores the ammonia in the arms and one that stores the ammonia elsewhere. And so in this ammoniacal arm, you can see this is a honeycomb texture where all of this would be filled with ammonia. And then in this muscular arm, this dense gray structure, that is all of the muscle. So you see very different morphology, very different structures in these two squids, which are both ammoniacal in nature. So cranchiids specifically sequester their ammonia in their coelom, so in their mantle cavity. This is full of ammonia. Batatoothidae uses dermal cushions, so on their skin, that's where the ammonia is sequestered. And then other species use it in their arms and in their mantles. So these are just some of the examples of sequestration methods, but they're diverse. Not only are the methods diverse, the squids themselves are diverse. So we have anywhere from a small little 20 millimeter squid to the biggest squid, the giant squid in family Architeuthidea. Both of those are ammoniacal squids. We've got these weird ones with these weird tails here, the strawberry squid, which is my favorite. So all of these are pretty morphologically diverse. In addition to this, we have Bathyteuthis. Bathyteuthis is a genus of deep sea squid falling in that clade Bathyteuthoidea. And it doesn't use ammonia, but it uses some unknown ion. We don't know what it is. Somebody was just like, this isn't ammonia, this is weird, and didn't go any further. And that is stored intracellularly. So with all of these unique storage mechanisms, unique morphologies, There has been two hypotheses as to the evolutionary origin of this trait. Is it a single origin? Is this a complex trait to evolve? It's only evolved once. Or are there multiple origins, which seems to fit with the more diverse morphology? But nobody has tested this molecularly. So that's where I come in. I used 165 egopsids and 5 outgroups, and I only used mitochondrial genes because these were the genes available in GenBank, ATP6, CO1, CO2, and ND5 from GenBank. I built my trees using Bayesian analyses in BEAST. In addition, I also used a divergence time estimation, but I didn't really do much with that in this talk. And then I constrained this tree by Fernandez Alvarez et al. 2022. They had a genome skimming tree for most of the families of Egopsida. And because we only have these single mitochondrial genes, I wanted a tree with more data to kind of constrain that topology and get rid of some of the mismatching that comes when using single genes. My ancestral state reconstructions I did with Bayesian analysis in Bayes traits using the BTW package in R, which is a really nice wrapper for Bayes traits. And then maximum likelihood analyses, I did that in APE in R. Because I talked about how ammoniacal buoyancy is good for deep sea species because it doesn't compress with depth, I hypothesized that this trait might correlate with the depth distribution of these ammoniacal species. And so I characterized each species as shallow or above 200 meters, deep or below 200 meters, or both spanning from the surface to the depths. And then in order to test, and I did the same ancestral state reconstruction as ammoniacal buoyancy. In order to test for correlation between these two traits, I used a phylogenetic generalized linear model in the phyloGLM package in R. So here is my tree. This is the result for our maximum likelihood analysis, and it shows 9 independent origins of ammoniacal buoyancy throughout egopsid history. Just to note, the results are the same for our Bayesian analysis. It seems that our egopsid ancestor, which is somewhere around here, it's kind of hard to see on this tree, is non-ammoniacal in nature. So this seems to have evolved post the evolution of egopsida. Naturally, when you think about this, you think, what's the alternative hypothesis? That single origin, what is the likelihood that could have occurred? How much better is this tree than that alternative hypothesis? And so the multiple origin had a likelihood of -58, and the single origin had a likelihood of -63. In this case, closer to 0 is higher, it's a better value. So there is a pretty large difference in the likelihood showing that this multiple origin is probably the correct hypothesis. Now we're going to look at our depth tree. So in this case, this is also the maximum likelihood analysis. Results, again, are the same for the Bayesian analysis. And although we can see, it visually correlates, right? You can see there's a large green clade of ammoniacal buoyancy here. There's a large green clade of deep right here. Same with this down here and over here. However, the p-value for our phylogenetic GLM was 0.9. So this is not a significant correlation. And I think one of the issues here is that we could only correlate things as shallow, deep, and bold. We don't have fine enough scale detail for most of the species in this tree to be able to treat depth as the continuous character that it really is. Most of these squids, egopsids, are highly understudied because a lot of them are not of fisheries interest. And so we either know it from one specimen, or it's like we find this occasionally on the surface of the ocean. We don't really know the ecology of a lot of these species. So although it looks visually correlated, there is not a significant correlation at this time. There we go. Okay. So overall, ammoniacal buoyancy has probably evolved multiple times in egopsids. Depth may correlate with buoyancy, but we need more information. As of now, it does not. And then there are 3 genera and 1 clade that need further investigation. So Magnapinna, Narratoothis, and Philidotoothis are 3 genera that we have no data for, but they fall in an otherwise ammoniacal family. So based on all of its closest relatives, they probably have ammoniacal buoyancy. And then the inoculatoothed families, there are 2 genera in the inoculatoothed families that have ammoniacal buoyancy, and then the rest don't. And so it's interesting, are these guys unique within that family? Are we missing something in the rest of the species? What is going on there? So there's definitely further research that needs to occur. So we've talked about Decapodiformes, we've talked about Egopsida, and finally we are moving to Pyroteuthidae. Pyroteuthids are small-bodied pelagic squids with little fisheries interest. They are really important ecologically as both predator and prey, as most cephalopods are, but they also are vertical migrators. So they live in deeper waters at night, around 300, 400 meters, and then they're moving to the surface during the day, or I mean, vice versa. They're living deeper during the day, and they're moving to the surface at night, presumably to feed, avoid predators, things like that. But they're providing an important energy connection from deeper water where all of the energy sinks in the ocean. It's produced at the top and it starts to sink below. So it's bringing some of that energy back up to the surface of the ocean. These guys are globally distributed. So that's there's some really interesting biogeographic questions we can ask here. And that is what I did. There are 2 genera. We have Pterygioteuthis and Pyroteuthis with 7 species. So present and future niches is where I'm going to start. So I did some species distribution modeling. I downloaded all of the Pyroteuthidae observations from the Ocean Biodiversity Information System, or OBIS, and I used all species with at least 10 observations. So unfortunately, Pyroteuthis serrata only had 5 observations from 2 localities. So those were not included. I was not able to do any of the modeling for that species. For environmental layers, I downloaded those from BioOracle, and I used surface values for temperature, current velocity, depth, salinity, dissolved oxygen, and primary productivity. I built my models with SDM Toolbox as prepping, so rarefying points, things like that, and then Maxent was the actual model building. Maxent requires background points, so I selected those using a bias file. And the reason I did that is because of sampling bias. This image you can see here is a map of the globe, and all of these points throughout are places where Egopsids have been sampled. They're Egopsid observations from OBIS. And you can see this sampling is highly concentrated along the coasts of the major continent. And we have some vertical tracks where it looks like it was probably a single expedition that just went up. And so I selected my bias points, heavily weighting the middle of the ocean and where there's fewer observations, and down weighting the areas where there's more observations to kind of hope to get rid of some of that autocorrelation. I evaluated each of my models with the emission error rate for the 10 percentile training presence. And then if that was the same, I looked at the area under the receiver operating curve, or AUC. I also wanted to see how these species are going to do in climate change. So I projected each of my models into the future using the 4 major representative concentration pathways. These are emissions scenarios, starting with 2.6, which is the best case scenario, and 8.5, which is the worst case scenario. And I did this for 2040 and 2090 to 2100. So here are present day SDMs for our Pterygioteuthis species. Yellow represents high habitat suitability, and blue represents low habitat suitability. Pterygioteuthis gemata and Giardia are global species, as you can see. Pterygioteuthis hoylii is concentrated in the Eastern Pacific, and then this is the Western Pacific. So right here, I think is Hawaii, and over here would be like Australia. This is that kind of region of the world. So most of the SDMs performed pretty well. We have 0 OERs across the board. You want your OER to be as close to 0 as possible. However, they weren't very discriminatory, which means they had a hard time determining the difference between presence and absence. And this can happen with animals with a really wide distribution, like in these globally distributed species. Except for Pterygioteuthis microlampus. Something bad happened here. I don't like this model. This is what it gave me. It has an OER of 0.9. Remember, I said it wants to be as close to 0 as possible. So this is a bad model. And then our AUC, it's higher than the rest, but still that OER is pretty high. And so I'm using this model for the rest of the analyses, but we're going to take everything I say about Pterygioteuthis with a grain of salt from here on out. Now for Pyroteuthis, again, pretty low OERs, great, but also low AUCs, which isn't ideal. Pyroteuthis adalux is living in across the Pacific, but really concentrated in that Western Pacific. And then Pyroteuthis margaritifera is globally distributed, but it seems to have a pretty anti-equatorial distribution. And this has been noted for some of the species. You can see it here too. There's kind of this swath across the equator of low habitat suitability. So this has been noted before, but it was cool to see that reflected in these models. So one explanation for some of these models is looking at current velocity. We're going to look at this where yellow represents high current velocity and blue represents low current velocity. So you can see the major ocean currents. You have the Gulf Stream here, the equatorial currents here. If you compare this model to Pterygioteuthis gemata, you can see low habitat suitability in the Gulf Stream, low habitat suitability across the equator. Note right here, over here off of the east coast of South America. We're going to look back at this again. So a lot of those low habitat suitability areas are concentrated on these areas of higher current velocity. So that was really interesting to find. Now to future predictions. So dark blue represents lost. So where the species lives currently and where it will no longer live in the future. Light blue represents where they stayed absent, so they don't exist now and they will no longer exist, and they still won't exist in the future. Green represents stayed present, so they currently exist and they will exist in the future. And then yellow represents gained, where they don't exist now and they will exist in the future. So every single species except for Pterygioteuthis microlampus gained habitat. Approximately 10% or higher, they're gaining habitat across the globe. There's a poleward expansion, which is expected for a lot of marine organisms, but we're also seeing expansion along the equator, which is really interesting. Here's down here in the equator. So I'm not exactly sure what's going on here, but it's the same for our Pyroteuthis species. We're seeing a lot of habitat that is gained across the globe. And both of these are for the worst case scenario. So they seem to be doing okay with climate change. However, and I'll talk about that in my conclusions. So methods, I also did some niche comparison. I wanted to see these species are sharing a range. Are they also sharing their ecological niche, or are they utilizing different environmental variables within that range? So I used Humboldt in R. And this is a cool package because you can quantify niches in geographic space, so on a map, and then you can also quantify your niches in environmental space. So just the suite of environmental variables that these species like to live in. And so what this package does is it uses a niche overlap test, which is comparing all accessible e-space. So combined the e-space of both of these species, do they have the same niche? But then it also uses a niche divergence test, and this is shared accessible e-space. And so what this says is, when looked at the values that these two species share, do they have a divergent niche? It's not just if they're living in two different spaces, they have access to two different environments, they're going to share two different niches, or they're going to have two different niches because they have access to two different environments. When given the same environmental variables, do these have two fundamentally different niches? So 4 of our species pairs, only 4, exhibited significantly divergent niches. And so I'm going to go through those now. So what you're seeing is the E-space for species 1, those environmental variables, and the E-space for species 2. When overlapped, you can see that it doesn't really look different, but what probably happened is that this is such a large, way bigger E-space than species 2, which is Microlampus, that when resampled, more often than not, they were getting values that were different. So just based on the size alone, these two have divergent niches. Pterygioteuthis coylii and Microlampus, you can see much better in this graph. Those hot spots are different. They're not overlapping, so they're not sharing a niche. If we remember, Pterygioteuthis coylii lives in the Eastern Pacific and Microlampus is living in the Western Pacific. So they don't have an overlapping range, so it makes more sense that they're having different niches. Pterygioteuthis coylii and Pyroteuthis adelux, again, there's this red hotspot here. It's vaguely overlapping with the blue, but the blue has more over here, showing different niches. And then Pterygioteuthis gemana and Pyroteuthis margaritifera, they have, interestingly enough, a similar shape environmental space. But when you overlap, those hotspots are just slightly off from each other. And so this is what that significantly different niches. They're not overlapping at all, even though the greater environmental space is. So overall, SDMs were not very discriminatory. Maybe this is because they're globally distributed, but people have hypothesized that there are some cryptic species in these global species. And so if we're including the niches from multiple species, it could lead to a lack of discrimination. It seems that pyroteuthis are going to fare well with climate change. However, this does not include ocean acidification. It doesn't include fishing pressure, and it doesn't include species interactions. So just based off of these things, and temperature is included, so it seems like they're going to do okay in the warm oceans. Climate change is going to be okay for them. But, and that's a big but, there's other factors to consider. And then finally, is there ecological speciation happening? Are these species having different niches? And the answer is, I don't know. 3 out of 4 of the species shown in those comparisons, they share a distribution. So there's something going on with those different niches, but 2 of those species included the bad model, the Pterygioteuthis microlampus. So I don't know if that E-space is categorized correctly. I'm not exactly sure if that's. And also there are only surface variables. So even these squids, remember, they live from the surface to around 300 meters. And so by only including surface variables, we're only getting a short snapshot of their niche. We're not experiencing the whole thing. So maybe there's some differentiation going on in deeper waters. Now on to the historical biogeography of these squids. So I used a Bayesian time-calibrated phylogeny in BEAST. And for these squids, unfortunately, I could only use CO1 and morphology. The single gene topology for this family were conflicting with every single gene. And you may ask, how do I know if this is the correct tree? I don't really know this is the correct tree, but CO1 had all of the species represented. So I had to pick that gene in order to actually include all of the species. And these are just baseline analyses. So if the tree isn't 100% correct, I can redo these in the future. We'll figure it out. But for my historical biogeography, I use biogeobears in R, with the extant regions being Pacific Ocean, Atlantic Ocean, Indian Ocean, and Mediterranean Sea, and also combinations of the two depending on where they live. And then I tested all 3 models available: dispersal, extinction, cladogenesis, diva-like, and Bay Area-like, and then plus jump parameters with all of those models. A jump parameter allows for a founder event, which isn't as common with these pelagic free-swimming marine species, but I wanted to test it anyways. So here is my tree. It seems, so Pacific is yellow, Pacific Atlantic is blue, Pacific Indian is pink, Pacific Atlantic Mediterranean Indian, so essentially global, is green, and then blue is this other. That other category is anything that had less than 10% chance. I just added it all up and counted it as other, so our pie charts looked prettier. It seems that the ancestor of Pyroteuthis is from the Pacific. Again, this is a pretty big other slice, but Pacific is the most likely scenario based on this pie. There are 2 independent dispersals out of the Pacific, one happening somewhere between 130 million years ago and 30, 50, I can't. Yeah. Right here when Pyroteuthis margaritiferus splits off. and then one happening somewhere around 50 and 6 million years ago. And this makes sense when we consider the paleogeography. 130 million years ago, when this family was evolving, the Atlantic Ocean was just a tiny sliver. So just by sheer probability alone, it is more likely that this family is going to have evolved in the Proto-Pacific Ocean, because that's just taking up most of the globe. We're moving to 80 million years ago, and these are the time slices where dispersal could have occurred. They could have come through the Proto-Caribbean Sea into the Atlantic. You've got the Southern Atlantic is forming now, come through the Drake Passage, Tasmanian Gateway, and also through the Tethys Sea, where the Mediterranean is, it used to be open, and this is a major gateway for animals into the Atlantic Ocean. 30 million years ago, it's starting to look more like Earth. Again, you still have the Proto-Caribbean Sea is open to the Pacific. All of these passageways I've talked about before, still open. You look at 6 million years ago, we have the closure of the Tethys Seaway. So if it happened, that dispersal into the Atlantic happened 6 million years ago, probably not through that Tethys Seaway. This is getting closed, and then these are still open. So there are multiple pathways, but some of them are less likely than others. And then I did some phylogenetic niche modeling. Have the ecological niches evolved over time? We know their geographic ranges have changed, but have their ecological niches changed? So I used phylloclimb in R, which takes single variable SDMs. So a species distribution model made only for current velocity, made only for depth, etc. And the phylogenetic tree, which produces these predicted niche occupancy profiles. Since these are only taking into account a single variable, I wanted to create PCs, principal components, to represent the entire niche. It's not an ideal way to do it, but it's going to allow me to take into account multiple variables at once in these models. We saw some divergence between species, so I wanted to test if that divergence was significant, and I used simulations in PhyTools with the differences between the traits for each species pair as the starting point. So here are our phylloclimb results. We have current velocity, depth, dissolved oxygen, salinity, temperature, and primary conductivity. All of these have time on the x-axis, with present day being the first year. And so what these dotted lines are the present day niche of these species. So this represents the current range of environmental variables that each of these species exist in today. And so you can see across the board, they're all overlapping. So they're all have an overlapping niche, probably not significantly different, which is what we saw in the niche comparison, except for those 4 species. But the means, it seems like, are diverging in some species pairs across the board. So it seems like there is some differentiation. It just hasn't reached significance yet. When we look at the PCs, you get the same thing. So PC1, the percent of variance is 38.67, and the percent of the variance in PC2 is 23.6. And you see the same thing. You see Gemata and Giardia are diverging. So sister groups are diverging pretty differently recent in time in PC1. You see some divergence in our Pyroteuthis taxa, but none of these are significant based on our phylogenetic GLM, or not our phylogenetic GLM, based on our simulations, which you can see the p-values down. And then, so overall, it seems like this family originated in the Pacific Ocean. There were 2 independent dispersals, one between 129 and 30 million years ago, and then one between 53 and 65 million years ago. Could be the Tethys Seaway, the Drake Passage, the Proto-Caribbean Sea. There are a lot of entrances into the Atlantic out of the Pacific Ocean. It seems that there is no significant divergence in evolution in niche space. And it seems like, yes, there is sympatric speciation possibly going on with these species based on that historical biogeography, but it seems like it's not happening through ecological speciation. Compared with that niche comparison plus these phylogenetic niche models, we're not seeing a ton of ecological speciation. Again, this is only the surface variables. So maybe if we get better rasters get better environmental data for those deeper layers of the ocean, there would be some more speciation, some speciation mechanisms found. But based on surface variables, it seems like there's some other way that they're diverging from each other. Our overall conclusions, that brings me to the end of my presentation. So RNA editing or data set size may affect phylogenetic inference. Ammonitical buoyancy has multiple origins. Pyrotuthids may fare well with climate change, but we need more data. And then most species seem to share niches. There is a Pacific origin of Pyrotuthidae and little to no evidence of ecological speciation. Overall, for Decapodiformes, but specifically Egopsids, more sampling is needed. There's a lot of these weirdos that live in the deep sea that maybe we have one video of them or one specimen, and there's just not enough data, or they're fixed in formalin, so we can't access them for genetics. There's a lot of data that we still need to collect out there. But the future of cephalopod research is looking promising. There are new technologies every day to access deeper waters. There is new sequencing technologies. Things are getting cheaper. So it seems to be a good time to be a cephalopod biologist. I would like to thank my advisor, Andy, my committee, Jason Brown, Kurt Newbig, Guillaume Bastille-Rousseau, Annie Lindgren from Portland State, who's on Zoom, and then the SIU Big Dog Research Computing Cluster, where I did a lot of my analyses, current and former members of the Anderson Lab, my Friday Journal Club friends, Conrad, Bree, and Taylor for being a sounding board for ideas and emotional support throughout this process. And with that, I'd like to take any questions.